Yeah, guys, don't worry. I'm just on unmute because people are actively joining. So uh, I didn't not yet started talking. Okay, we'll start the session now. So you guys hear me now? Is it clear?
Yes, sir.
Yes, sir.
Yeah, yeah, great. And you can also see my screen, right?
Yes, sir.
So yeah, that's good. So we'll jump onto the session today. First of all, I welcome you all for this course on advanced bioinformatics. Okay, so a lot of people have joined newly to this course, so I welcome you all. And yesterday we just had an overview of the syllabus, and we had a query session, like a brainstorming session. We had discussed a lot, and today we'll be jumping on to the first content. Okay, that is Introduction to Advanced Bioinformatics, and we'll be discussing about overview and advanced bioinformatics techniques and their application, role of software tools in bioinformatics research, emerging trends, and challenges in the field. Okay, so yeah, just a minute. Okay, so good evening all. Let's start with the session now. And I just wanted to give you some small disclaimer. Today, today onwards, we'll be like discussing the whole part altogether. And if you guys have any queries, you can put it in the chat box, or else you can just write it down and save it for later, so that after the session, we could just discuss about that. Okay. So in between, we can just uh, stop and we can just discuss about the questions. So a lot of people have mentioned, because since we have only 1 hour time, we'll just try that approach. Okay. Let the session go on a flow, and at the end, we'll have the query session. Is that okay? Is that good?
Yes, sir.
Yeah. Okay. So I'll share my PPT now. Okay. Just a minute. So do you guys could see my new screen, new PPT, new slide? Is it visible?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Yeah. So check. Okay, so let's start the session now. Let's jump on to the first topic, that is Introduction to Bioinformatics. So we'll just briefly discuss about what is bioinformatics and advanced bioinformatics. Yesterday, I told the word that advanced bioinformatics, the word advanced here is nothing but, it's just nothing but we're just going to learn extra. We're just going to dive deep into the concepts of bioinformatics, and pretty much nothing is there. Like advanced, the word is just telling about something extra to learn about bioinformatics. And I'm your course instructor, Rajesh. And this is my LinkedIn ID. You can check it later. So, introduction to bioinformatics, bioinformatics. So, advanced bioinformatics is a multidisciplinary field where it combines biology, biotechnology, computer science, statistics, mathematics, and other closely related subjects to develop and apply computational tools and techniques for understanding the biological data, to play with the data what we have, and to annotate or to process the data. Okay, so that is what advanced bioinformatics. So it goes beyond the basic analysis of genetic sequence to encompass more complex and integrative approach. We are going to do more than that, like not only playing with uh, only genetics or genomics part. We have a lot to do. Okay, so that is what we're going to learn in this course. So here we'll be talking about multi-omics approach, multi-omics data, and integration of those data, and difference between structural and functional biology, and also system biological aspects. Okay, so what are the key areas of advanced bioinformatics? So, since this is a root of bioinformatics, we and some advanced concepts are there in the this kind of course. Just So, first thing, there are a lot of key areas where we can just explore on, and those key areas are given here. First one is sequence analysis, where the analyzing of sequences of DNA, RNA, or protein, and to identify its pattern, how this particular sequence, DNA in the form of gene or genome, RNA in the form of different types of RNA, the miRNA, siRNA, like those kind of RNA sequences, and proteins. We have different foldings of proteins. We have different structures of proteins, primary, secondary, quaternary structures of proteins. And to identify its pattern and similarities and functions all put together, we'll do this all these analysis in the sequence analysis. So here the data will be in a sequential way. Okay, all will be like in case of DNA, it is consist of A, T, and G and C. Okay, and in the case of RNA, we'll be having uracil in like in the place of T's. And protein structure, this single letter code of each amino acid residue put together to construct a protein. That is three amino acid uh, codes for a protein. Like that's the protein sequence. So application is identifying gene functions. Especially, we can deeply dive into the concepts of genetics with the help of this in silico analysis, and evolutionary relationship like phylogenetic tree analysis between cross variants between different organisms, like that. It is helpful. And predicting the effect of genetic variations. We'll be having genetic mutations, genetic drifting, so that it is easy for us to study while analyzing the sequence analysis. It is easy for us to study how far the genetic variation happened and where all the places where this genetic variation happen. Okay, so sequence analysis is one of the key area. And next thing is genomics. Genomics is one of the omics approach which we play with genes. Okay, the study of the complete genetic material called a genome. So collection of genes are called genome of an organism. So that will be comes under genomics. And the application is genome sequencing, genome assembly, comparative genomics, and functional genomics. So this is a pretty basic slide because I just wanted to start the easiest way, because the upcoming classes will be so maybe so outlandish, or we'll be having dealing with a lot of inputs. So in this slide deck, I've given some links also. In the last few pages, I've given some links for the resources, for the tools, especially. So what you're going to use. And next key area is metabolomics. This is also one of the omics approach of metabolic studies, like the study of chemical process involving in production of metabolites of an organism. It can be a small molecule substrates, or the intermediates between the process, the biological process, and the production of metabolites and metabolism. Like organism metabolizes everything in order to survive, right? So that all comes under this computation part called metabolomics. Okay, so The application part is understanding metabolic pathway. Yesterday we discussed about a lot of pathway analyzer, pathway integrators called KIT pathway, ITOR, like that, and disease mechanism. We can identify how a disease is proliferating or acting upon an organism, where genesis and how pathogenesis happens, and the discovery of new biomarkers can be developed. Like with the help of metabolomics, we can develop new biomarkers for respective diseases, like for cancer, for various types of cancer, etc. Like that. Next is epigenomics. So it is the study of hereditary changes in a gene. So the hereditary changes where comes when a whole set of mutation happens in a gene, and those changes will not change the DNA sequence. There will be no changes in the DNA sequence, but we can see a remarkable change in the genetic marker. So that is called epigenomics. The study of that is called epigenomics, and investigating the DNA methylation process. DNA methylation, or these are DNA alteration. You can otherwise it is called as DNA alteration methods. Okay, investigating DNA methylation, histone modification, and their roles in the gene regulation diseases. So ultimately, guys, uh, you know very well about central dogma. So uh, like DNA to RNA and RNA to protein, and it comes all the way around, like reverses. So we will be dealing with DNA, RNA, and protein a lot in this bioinformatics. Okay, we will be playing with those kind of data here. So uh, this is one of the key areas where we could thoroughly learn about the modifications happening in DNA. That is both artificial and natural mutations. Okay. So, and next is transcriptomics. This is the study of a complete set of RNA transcript, like when produced by a genome under specific circumstances, when some kind of mutation happens or some kind of bodily activity happens, when cell signaling pathways interferes, or some kind of interference happens with the interaction between the cells or interaction between the molecules. So and that time, this RNA transcripts will be produced. Different types of RNA transcripts will be produced. So it is the specific part to study about the transcripts, RNA transcripts, when it comes to genome studies. Okay, and that part is called transcriptomics. And next is the best part, the proteomics part, where we will literally play with proteins and protein associated compounds like enzymes, cofactors, and other stuff like that. This is the large scale study of proteins, including their structures and functions. So each protein have a specific structure. Each protein have their specific function. Okay, and behind that, you'll be learning about the amino acid residues, amino acid sequences, and the bonds between them, and how those bonds like influence the structure and their functions. So identifying protein interactions and post translation modification. You'll be hearing this a lot, post translation modification. So this will be helping you in disease identification. Post translation modification is the primary cause of something happens when disease is proliferating. Okay. So Arul, you raised your hand. You want to ask something? Arul Narayana. Okay. So, and we can also identify disease biomarkers using this proteomics studies. Next thing is structural bioinformatics. Okay, the name itself suggests that we're gonna annotate, we're gonna visualize, we're gonna deal with structures. Okay, different structures like three-dimensional structures of biological macromolecules. Here, macromolecules are always DNA and protein. Okay, so to analyze and predict the three-dimensional structures, 3D structures. Okay, there are a lot of tools, a lot of resources where you analyze the three-dimensional structure. You can download it. You can literally view and modify, or you can even do some changes to the molecules inch by inch. Okay, like this all plays a major role in drug designing and understanding the protein functions. Like this is one step ahead. Ahead to the proteomics, like understanding protein functions and molecular interactions. You'll be going to the molecular level, and you will be seeing how these interactions are happening. What molecules, like how a protein interacts with a protein, or how a protein interacts with a ligand. Again, in the sense a chemical compound, okay, or it may be a drug, or it may be a drug, or it may be a chemical compound which is you isolated from a plant or animal source or anything. You purified it, you isolated it, and that can be a ligand. Okay. Next thing is system biology, an integrative approach to understand the complexity of biological systems. So we biological organisms, we are so complex. We are made complex. Okay. So one of the approach, that is the mathematical computational approach to decipher and to break it down, the complexity of this biological system is called system biology. So under this, where all we use the system biology is on network analysis, pathway and modeling, and studying the dynamic behavior of the biological systems. Like dynamic behavior in the sense, like both functionals and structures, and also the eccentric part of this networks, biological systems. Okay, like even starts with a simple cyclic pathways, and all the way up to complex, intermediate, and yeah, intermediate and put together systems. Okay, so we'll be seeing some of the tools what we are gonna use this bioinformatics research, or what we're gonna commonly discuss in this particular course. Okay, so bioinformatic under bioinformatic tools, there is role of some tools. First thing, we always look into this data analysis. So when it comes to a computational biology, or when it comes to a computational method, anything we look into we look into it is a data. Okay, anything we call it as a data. It may be a raw data, it may be a process data, it may be a data which is acquired from in vitro process, it may be data where it can be acquired from a in vivo or in silico, whatever it may be. So in end of the day, it is a data. So first thing, we'll give importance to data analysis softwares. So most of the softwares, like softwares and frameworks, what we're going to use here for as a tool, or as an analyzing module, is our programming, Python and bioconductor. So data, in the sense, you don't just think of some graphical data. The data which I'm telling is here, it's kind of something, a plot kind of data, plot, tables, graphs, like that. Okay, that kind of productive data, which has qualitative and quantitative measures. Okay, and the applications are statistical analysis, machine learning, and also data visualization. So machine learning and data visualization, these particular frameworks plays a major role, and we'll be slowly learning about these machine learning algorithms and data visualization tools, other data visualization tools, other kind of graphical user interface tools. Okay, next thing is database management. So when it comes to bioinformatics, we usually deal with a lot of databases. Okay, we have different databases. For example, we have NCBI database. It's a repository of whole organisms, uh, like biological molecules and biological systems. Okay, we can analyze, or we can harvest a lot of data from NCBI itself. And some of the secondary databases we have for protein, we have for drug, we have for chemicals. So we have a lot of databases. So database management tools are given here, like some examples like GenBank, and Ansible browser, genome browser, and ULCAC genome browser. So these database management tools are used for uh, us to store data or retrieve data, and to process in such a way where it will be helpful for us in later analysis or later research studies. Okay. So yeah, Krishnamurthy, you raised your hand. Am I
Sir,
audible? Like, you guys could follow
yes,
me?
sir.
Am I audible? Yeah.
Sir,
Yes.
hello.
Yeah. Yeah.
Yes,
Yes,
sir.
Krishnamurthy.
Sir, what is the purpose of the gene bank database management means for maintaining the gem plus information sources like assassin ID or like entire gene
Yes,
bank?
yeah. It is kind of
Yes.
plays a both kind of role, where you also publish data, and you also can access data of people who published their data. Like they'll be doing research, they'll be publishing that data in a public domain, where you can read about it, you can take that for your research, you can analyze it, you can annotate it. So that is what GenBank is. Okay, GenBank is where you submit
Okay.
your genetic data, like you'll be sequencing using 16 as RNA sequencing, 18 as RNA sequencing, and whole genome sequencing, like that. You'll be publishing that in GenBank or Bankit. It is the resource of NCBI. So that is one of the database management tool. Okay, so it is managing, like it has the wholesome data of all the biological systems. That is what database management tools. Okay, we can consider this as tools, or we can consider this as some kind of engines maybe or frameworks. Okay, these are all different words. Yeah. Yes. So some of you guys having trouble in joining. Is that so? So my voice is not clear. What about now?
It's clear, sir.
Yeah. Okay. Okay. Yeah. Fine. This is the head count today. Yeah, it's a pretty good head count. Okay, we'll continue. And next thing is workflow management tools. Like you will be having a lot of workflow, a lot of pipeline in computational methods. Like you can't sit with the hours and hours with the annotating or while analyzing a data. We'll just in a specific time frame we'll just lose hope, lose our
Sir, your
interest
voice is
over
breaking in
thing.
between.
Am I audible now? Am I audible now? Is it clear?
Sir, it's
sir.
cracking in between, sir.
Okay. Yeah, I'll check my network. I guess I have small issue with my network. I'll just reset the settings. Uh, is it okay now?
Yes, sir.
Guys, good here. Yeah, that's good. So yeah, to automate that kind of workflow. Okay. So when you do something in lab, you have a procedure, you have a methodology, right? You'll be following that procedure. Or else, like let's consider a scenario where you work in a wet lab. Okay, you'll be following some steps, and that steps will be leading you or giving you a proper result. Okay, where sometimes human-made errors may happen. At that time, you'll be redoing the job, right? So that is wet lab, so that we could rectify something. But on bioinformatics or defining a workflow in bioinformatics, it's a huge process. Like there are anyway, the next topic is NGS data analysis. It's a huge part. So we can't just sit with a laptop for hours and we can't just look into the data and we couldn't predict where the error happened, or we couldn't manage a lot of data, right? Like heavy size of data. And it should be automated. At certain point, it should be automated. It should be running actively with or without the knowledge of us, like knowledge in the sense overviewing of us. Like we'll be looking together the systems. So that kind of workflow management tools are Galaxy, Taverna, I say Apache software, Apache search engine software, and Nextflow. So these tools are helping in streamlining the complex bioinformatics workflow. Okay, so you'll all be doing this docking, molecular and docking simulation. Like when you're doing this blind docking, you have a set of process like protein preparation, ligand preparation, grid construction, and you'll be running the grid and you'll be running the docking and analysis and post analysis. So this is all predefined. So when it comes to molecular dynamics simulation, like I'll just give an example. So molecular dynamics simulation is where you do lot of steps, and even running of that simulation part sometimes even take days. Okay, and you can't just sit in front of the laptop for days to get the data, to get the result. So that kind of workflow should be automated. It should be running at the back end, and that kind of process can be streamlined on these three softwares. Okay, Galaxy, Taverna, and Nextflow. And I've given these links on the last pages of this particular slide deck. I'll share the slide deck with you. Don't worry. Next is visualization tools. So any raw data should be processed, it should be defined, and when it is provided to the scientific community, or when an active researcher is taking that data and wanted to publish it, should be in a form where even a layman could understand it. Layman could understand the result, or he could understand or interpret the result, right? So it is so crucial that the visualization tools are playing major role in helping to understand the raw data, or in the conversion of the raw data into a presentable data. Okay. So some of the examples are Cytoscape. You see as a camera, ggplot. Ggplot is one of the R package, R programming package, and plot is also one of the R package. Okay, so this helps in creating interactive and informative visual representations. So when you go for publications or book chapters, so we can't just run around some screenshots, random screenshots, or we can't just run around the manually created table. Okay. Sometimes you should create a data or visualization representation which interacts to the viewer or the reviewer or the person who reads the article or the person who overcomes or the person who refers your article, right? So that is more important, creating interactive, and it will be much more informative. It should be much more informative. So creating that kind of data is one of the crucial role uh, in bioinformatics. So we'll be learning about that visualization tools also. And next thing is emerging trends in bioinformatics. So the normal bioinformatics, what a couple of years or some decades before, is not the bioinformatics today. Okay, so the bioinformatics today is totally different because today I mentioned the word like these computer science people or technological people, they are too much like they are very ahead of us. Like they are doing a lot of things, they are creating, they are automating a lot of things, and we biology people also do that kind of stuff. So this is one of the approach where we can compete with those people. Like we also take equal part in the technological aspects. So one of the aspect is called big data and machine learning. So usually bioinformatics is a generic a person. If I call a person from a biotechnology background, if I ask what his bioinformatics is, he'll tell, yeah, sir, this is NCBI, and sir, this is RASMOL, sir, this is Uniprot, and we can visualize data and this like that. So, but bioinformatics is totally different. This advanced bioinformatics is totally different. So, have you guys ever heard of the tools just I told before, like I told just now in the previous slides? Have you guys heard those kind of tools?
No, sir.
So,
Yes, sir.
Actually,
yes,
this is for the first time.
yes, some of you may heard, some of you may not. And most of you guys are from biotech background. I mean, even the course which your college is offering you won't talk about this much, this kind of tools and this kind of techniques much. Okay, because the first myth or the first misconception what we have, even well-educated people or well expert domains in this particular life science, is they see this bioinformatics as a dry subject, something called a dry subject. Okay, so this is not first of all, you don't see it as a subject, this is knowledge, and next thing is this is not dry. Okay, so it is literally like playing a game, playing a computer game, where it'll be so interesting to analyze, visualize, and process the data. And yesterday I told you you could just publish a good quality paper with a good setup laptop and some knowledge about some tools and a proper approach. So that is what bioinformatics is now. So big data and machine learning are the emerging trends in bioinformatics. So leveraging large data sets and advanced algorithms to uncover new insights of biological research is what done by big data analysis and machine learning. So application includes predicting models, like creating predictive models or predictive modeling, and personalized medicine. So personalized medicine also emerging trends in life science, in pharmacy research. So, according to the person's stature, according to the person's medical profile, the medicine can be personalized and administered. So, we have n number of diseases, we have n number of conditions, we have n number of traits when it comes to genetics. So, in that case, the biological data is abundant. It is more. So, as I told you that the data what we have or what we or for example the human genome project, it is having petabytes, sorry, terabytes and terabytes of data. Okay, so that kind of data only be analyzed or only be approached only with big data analysis approaches. Okay, and data mining. Data mining is where you with a single click of a button or passing a command with a single passing command, you can just analyze or you can harvest or you can cite data what you need, like GBs and GBs of data. Next thing is single cell genomics. This is a technique to study genome and transcriptome at the single cell level. Okay, so not in a tissue level, not in the multiple cellular level. You can study genomes and genomics and proteomics at the single cellular level. Okay, you take a cell, one single cell, it may be any kind of cell, and you can check its genomic and transcriptomic properties. And there are several techniques to do that too. So application includes understanding cellular heterogeneity and developmental process. Like all the cellular processes are different from each other, and they also have connection between them. Okay, so when we talk about this KEP pathway analysis and all, if we go to this disease section, we can just see what are the pathways, single pathway that involves an n number of diseases. So likewise, we have a lot of biological process involved together, or some kind of molecule that interacts and alters or modifies some action at the back end of a biological process. So everything will be put together, and we can apply this particular techniques on those aspects, like in development processes also. So next thing is CRISPR and gene editing, CRISPR-Cas9, CRISPR-Cas12. So we have heard about it. Anyone have any idea about this CRISPR technology, gene editing technology, gene silencing, gene editing?
No, sir.
Huh? Okay. So it is where you analyze, or it is where you take take a gene, you can modify it, you can edit it, you can snip and remove the unwanted sequences like that. You can modify that genes. Okay, that is what uh, CRISPR and gene editing is. So bioinformatic tools for designing and analyzing CRISPR experiments. So CRISPR is not a bioinformatics approach. CRISPR is a wet lab approach, but we should have some bioinformatics knowledge to analyze or to design the workflow of this CRISPR, of gene editing technology, because we have genomics. Okay, we have functional genomics. Like we have separate topic on genetics and genomics, right? So that will be helping in designing the structure or designing the process of CRISPR. Okay. So the application includes precise genetic modification and functional genomic studies. Sorry. And next is metagenomics. So where multiple set of or multi-organism genomes are put together, analyzing in a single column or single set, or converting it to a single set of data. It's metagenomics, like analyzing genetic material recovered directly from the environment. So that particular environmental sample will be having n number of genetic materials from different organisms. Okay, so normal flora and fauna will be having a number of bacteria, a number of fungi, or a number of parasites maybe. So all this have their genetic material. So when we take a sample from, or maybe a swab sample, we analyze that in the wet lab and we come up with the data. That particular genetic material or that particular genetic marker will be different from the purely available thing. Okay, so that is what a collection of genetic data or genetic marker put together from environmental sample, that is called metagenomics, like analyzing those kind of samples called metagenomics. The application includes studying microbial diversity. So microbes, we can't just say microbes are microscopic organisms and so, like generic stuff. Microbes are far more than that. Okay, so if you guys are being microbiologists, you guys know about microbes, right? Microbe diversity. What are the species we have? What are the genus we have? What are the strains? Different types of strains and substrains we have. Even E. coli has around 121 different strains, right? Like that. And ecological interactions between those microbe diversity can also be analyzed using this metagenomics approach. Okay. So next thing is, yeah, challenges in bioinformatics. So when it comes to challenges in bioinformatics, so first challenge as a bioinformatician, what you face is managing and analyzing the enormous amount of data. So data which is available as of now, this year or this particular timeline, what we have here is enormous, is gigantic. Okay. So can anyone tell when this human genome project started? Genome project.
1984.
So at that time itself, yeah, very good. And that time itself, the Human Genome Project have started, and it went for around another 12 years, I guess so. So
Sir, I think it was
it
I
very-
guess
just a
2006 or
minute,
something.
just a minute. When it started, yes,
1990.
1990. Yeah,
Yes, sir.
you have conflict. You guys have conflict with you between you.
Yes, sir. In
It has started
1990, they
1990 only. It has started. Okay, not on
the
2006.
sequence.
That is what it just went around several years. Like it is completed on 2006 or seven, but it started on 1990. Okay,
Sir,
that much year it
Yes,
took
sir.
to complete. Yes, yes, yes. Someone have raised your
Sir,
hand.
they
Sorry,
identified. Yes, sir. That they identified and sequenced more than 30,000 genes in
30,000
human
genes.
genome
Yeah.
project. Exactly.
Yes. Great, great, great.
Yes.
Thank you so much, Krishna.
Yes. Yes.
And
Yes.
anyone want to tell something else? Yeah, Amit, you've raised your hand.
Sir, in the previous slide, you were talking about CRISPR. So
Yep.
would we be designing a workflow for a CRISPR experiment? And
Uh, you're asking
will,
whether we are going to do that?
yeah, or you are going
Oh.
to teach us how to do it and we can try
So as
later?
per the syllabus, we are not going to do that. That's no part in CRISPR. But when you're talking about this workflow management tools, so there is
Yeah,
also an option in Galaxy to design an experiment for CRISPR. Okay, and I'll tell you what are the tools that is also used in CRISPR. Okay.
okay.
So
Yeah,
if time permits or if we have some sufficient amount of time, we could just note this topic and we can just also discuss on that. Okay, later.
and sir,
You can
one
make a
more
note.
question I had.
Yes, yes.
Is there a Python package for assembling genomes or stuff like that, so that we can do that via coding?
You
Some,
want to automate it?
yeah, yeah, yeah.
Okay, okay. So what's your background actually?
Sir, I've done a master's in molecular biotechnology, and now I'm looking forward for.
So
So I'm looking at a composite, career going forward. Like I want to have little bit knowledge of bioinformatics as well.
computational.
Yeah,
Okay.
computation, so that the world is moving forward towards multiple experiments in the lab at the same time. So
Yes,
I want
yes.
to automate that
So
process.
when it comes to automation for biology or biotechnology, BioPython is the best package. BioPython has a number of libraries pre-built and a number of package pre-built. Okay.
Okay, okay,
Have a note on BioPython. There is something called BioPython, and its logo is also different from other things. Okay, there is a number of BioPython modules, and which you can just copy and you can grep it from. You know what is grepping, right? You'll grep it in
yeah,
Linux, and you can just install it in your local system or in the terminal, and you can just run it along. Okay, or
yeah,
else those some of the bioconductor and some of the BioPython packages available there. You can just use it in Google Colab also. You know what is Google Colab, right?
yeah,
Collaborative notebook of Google. So it's
yeah,
a code compiling framework where you can use it in web. Okay, so others please make a note. Google Colab or Jupyter. These are online code compiling frameworks. Okay, so you can
yeah,
compile Python codes, Perl codes, different set of codes, actually scripts. You can run those scripts on your web using your browser. You don't need, no need to need any IDE, or you don't need to need any sort of software, special softwares like Visual Studio Code, Node++, like that to run the code. Google Colab comes in handy. So I just wanted to add on some point. Previously, I just simulated a molecular simulator, molecular simulation, right? I simulated a molecule using Google Colab. So Google Colab is offering some GPU which can push, it's a virtual GPU, right? So it can push the process to its utmost limit and it can provide the data. So you guys know how tough is to perform this molecular simulation, right? But I'll just tell you something. You can perform this molecular simulation using Google Colab. Okay, there are steps for it. Okay, and don't worry if you're not having a good system or good laptop. You can also rely on cloud computing. You can go for AWS. Okay, or you can go for Google Cloud Provider Services, or you can go to Oracle. They have cloud computing platforms where you can run this workflow, or you can automate this bioinformatic pipelines through web in your cloud. So it will be not saved in your local system. It will be running, it will be having throughout internet connection and other necessary parameters. So it will be running in the backend. So you can retrieve the information and you can put that data in your research. You can include that data in the research. So bioinformatics is not only about the local machine what you have with you, the laptop what you have with you. You can also do bioinformatics and automation stuff, automating this bioinformatics workflow in cloud computing, through cloud computing. And you can very well you get the help of ChatGPT and other GPT platforms, AIs, to construct scripts. Okay, so it is one of the exciting part in bioinformatics. Yes. So Amit, you wanted to add on something?
No, sir. No, sir.
Yeah, thank you. So yeah, just make a note. Google Colab and Galaxy. I already told you Galaxy regarding Galaxy, and Jupyter Notebook is also one of the uh, Python coding platform through your web. You just make a note. You guys can note it down. Otherwise, if you want some notes or you want to include in the slides, I'll just include those links or some resources regarding those in the upcoming classes. Okay. So BioPython, you can also check on BioPython. Okay. So next thing is, so data volume. So we have abundant of data, right? So one thing is when it comes to a biological data, you should be running around the data and should be processing it very well in order to get some output. So the volume of data is enormous, and the keep this Human Genome Project in mind. It went from 1990 to 2006. Okay, so that is how this is the primary aspect what they was the primary challenge what even they faced. Okay, the volume of the data, or even now the volume of the biological data which is there in the open source repositories is so enormous. So what we can do is like solutions can include high performance computing. I told you like cloud-based computing, right? Cloud computing, you can rely on cloud computing or some kind of the based frameworks like this. Next one is uh, someone waiting in the waiting room, having this pop-up message from first. So okay, so next thing is interoperability. Interoperability is where like you have different databases, but you couldn't mark the interaction, or you couldn't mark the common things between those data sets. And the thing is, it is so diverse. It is so diverse, and there is no specific tools for even all the diverse kind of things. Okay, so integrating diverse data sets and tools which is used to do that to ensure seamless data in exchange of analysis is so tough, and even well-renowned or well-performing bioinformatician would struggle to identify the diversity between these data sets, and in identifying a proper set of tools and proper methodologies to uh, identify those outputs. So ultimately, it is going to be a computational outputs, right? So computational outputs will sometimes end up in errors. So that kind of errors may occur. So solution for this may be in developing standard formats and protocols. So only thing is, if a person who works in a framework or works in a workflow, like for example, we'll say a person who's analyzing NGS data, or for your ease, let me tell, if you guys are working in molecular docking simulation, so where you keep a protein and you keep a ligand, and you'll follow set of rules or set of steps. Okay, and those steps are predefined. But the thing is, you can't reproduce the same steps to get the same data what you got earlier. Okay. So the thing is, it should be the protocol. It should be pure. It should be predefined. And it should be well defined so that other upcoming bioinformaticians could use it, and they can reproduce. Okay. So that is what the major challenge. And next thing is reproducibility. Yeah, this is one of the major aspect. So we'll take this as an example. Same this phylogenetic tree construction and also this docking or simulation. When you run a molecule, when you complete a molecule, when you complete a blind docking, and when you run the confirmation for another 10 times, 10 confirmation, 10 different confirmation, and you'll be getting the binding affinity and ligand efficacy of the particular interaction. So when you do the same uh, when you do the same in a row with the same molecule, sometimes it may end up in giving different results. Sometimes it may give minus 9.5, and sometimes it may give minus 9.5, minus 9.6, or something like that. So reproducibility of this particular workflow is lacking there. Like uh, we can't expect the same result to be provided or to be produced while running the same set of procedure over and over again. Okay, so ensuring the bioinformatic analysis are reproducible and transparent. So transparent in the sense, how many of you guys just know to do a proper molecular docking or any sort of bioinformatic analysis? Like I'm talking to your UG level, like phylogenetic tree analysis or retrieving faster data or running a blast. So how many of you have a proper protocol or proper step to do this particular things?
Yes, sir.
If I guys handpick some of you and ask the same question to you guys, some will tell it in different way, different manner, and different. Yeah, a lot of people have raised your hand. Yeah, Tejul, just unmute and talk. No problem. Just tell me. So someone is setting their screen. Yeah, Vaishnavi, I guess you're presenting your screen. Okay, so this is what is happening. The reproducibility is lacking there. We can't just expect the same workflow to work over and over again and give the same result. Okay, so to avoid that, can you guys hear me? Like, is my voice clear? I'm
Yes,
facing
sir.
some network
Yes, sir.
trouble here.
Yes, sir. Yes.
It's
No, no, no.
Yes,
So,
It's
sir.
okay.
Yes, sir.
sorry if my voice is like this. Okay, so solution is to have a standardized workflow and version control to update the softwares. So pretty much all the softwares are built on Python, built on Perl, built on Java, and other equally important computational languages. So it should be equally, it should be updated. It should be constantly updated. Like how you update your WhatsApp or how do you update your phone, like that should be constantly updated. The bugs should be eradicated. The debugging process should be done, and security patches should be updated. So likewise, this particular bioinformatic tools also have this version controls and version updating thing. And this particular workflow should be documented properly. Okay, so every time you do a bioinformatics workflow, just document it so that we can rectify errors later, and it is also good to reproduce. Okay, and next thing is ethical issues. Ethical issues, not like not the ethical issues which comes on some kind of animal ethical committee like that. This is like addressing privacy concerns. So anything or any data is end of the day is a data of or it's information about something, right? So let's say we are just analyzing the genetic markup and we're going to identify a possible biomarker for diabetic type 1 patients. So we'll be talking to them. We'll be constructing a data set anyway, and those particular informations what we gather from them is subject to their privacy consent, right? So even though it is the data and we're going to use that data to construct something, or we're going to automate or do some approaches on identifying this in silico approaches. End of the day, those informations are those people, like they're individuals. And this data sharpening policies, data sharing policies and ethical consideration of this data is still a question. It's a questionable matter. So, how many of you have published or something in NCBI or GenBank or Bankit? You guys have any GenBank submission, something like that? Or you guys have ever heard of it? Like public submitting your yes, we'll share the PPT. Okay, we'll share the PPT. Have you guys heard of those kind of things, like submitting your 16 RNA sequencing in the public domain like that?
Yes.
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 So a lot of
Those.
people don't do that. A lot of people just wait until they publish that in a paper under their name, and after that they'll publish in the public domain for public use. Right. This is all happening. So data, whatever data we produce is not safe outside. Not safe in the sense, it's not like people will gather that information and they'll publish it and they'll work on. But the thing is, this lack of conviction will be there. And this ethical consideration is huge in the genomic research. Okay, whenever a genetic data or a genetic markup is identified or genetic glitch or anything is identified on the genomic level, this ethical consideration of doing that research on particular organism, for say example, analyzing a particular genetic markup of a P53 induced cell in a Swiss albino mice. Okay, in that case, the ethical consideration are so stringent. So you can't just carry out a genomic research, driving blood from a human being and just doing analysis and converting the raw data into a computational valid data. So it is not possible all the time. Okay. So the solution includes stringent data protection measures and ethical guidelines. So there should be, according to me, there should be one stringent guideline which uh, shared among all the life science people, so that all follow the same thing. And next thing is data protection. Like if you are providing a data, you are producing data that is intellectual to you. So you should be doing what is required to do, like creating a patent or creating a copyright or publishing it in a well-renowned journal. That can be done, right? So it should be in a proper manner. Okay. Next thing is, so yeah, I guess the presentation is over. These are some of the links what we talked about. Okay, so these are some of the links of the home pages of some of the tools what we have talked about in the previous slides. So first one is R programming. So we can click on this link and just visit the R programming. Next is Python. Python 3.0 is one of the best version to work with, and you can just download. And next is GeneBank. GeneBank we talked about GenBank here in the genetics part and genomics part. And Ensemble Genome Browser, there's a browser specifically to analyze, annotate, and to visualize the sequence of a genome and to identify the open reading frames and other aspects of a genome. And it is in a browser form. It is called Ensemble Genome Browser. And the same way, we have another browser called UCSC Genome Browser. These are so much important in genomics part. Next thing is this workflow, constructing a workflow or automating a workflow can be done by this Galaxy and Taverna, Apache, and Nextflow. You can click on this websites. You can just explore the options there. So when you go to Galaxy, you can see n number of bioinformatics workflow. You can just click on it with a single click of a button. You can construct a workflow, and you can just happily sit and watch what magic is happening there. Okay, so it is amusing to watch. Next thing is data visualization software like Cytoscape, UCSF Chimera, and ggplot of R programming and Plotty for R programming. Next things are Autodoc, what we use for this molecular dynamic simulation, molecular docking simulation, and FGL tools, a collection of tools from translational computational genomics diversity. And next thing is PyMol from Schrodinger with data visualization, that is 3D structure visualization tool. And OpenBabel, we commonly use this OpenBabel. It is a file conversion software. So you'll be converting files, right? PDF to document or Word to PDF or PDF to CSV like that. So in that way, we have a lot of biological databases, biological file formats. So to convert a particular file format into another. For example, when you download a chemical from a PubChem, it will be in SDF format. But when you wanted to prepare it as a ligand, it should be in PDB format, protein data bank format. So in that case, you will just go to this open label. It's a GUI, that is graphical user interface software. Can be downloading it and you can convert the file formats. There's a number of file format conversion. And next is BioWare Discovery Studio. T3s, sorry, 3Ds. Here you can run small molecule simulation. You can analyze the receptor ligand interactions. You can do molecular interactions. You can visualize it in 3D, 2D, and you can create a binding site. And you can do pretty much more. You can play with macromolecules with this BioWare Discovery Studio. It is used to visualize. Okay, and Schrodinger Suite is the premium package of bioinformatic tools. So I wanted to tell about Schrodinger Suite. It has different set of tools called Mastro, Desmond, and Pymol is also one of the Schrodinger Suite, but Pymol is free. Okay, and if you're being a bioinformatician, you'll come across this package or suite called XPC. So XPC is one of the Swiss Army knife. Yesterday I told you Swiss Army knife of a bioinformatician, where all or some kind of resources, like a lot of resources, will be put together in a single homepage, and you can just with a click of a button you can analyze or you can check or you can go and read its data. Okay, so XPC is the Swiss Army knife of a bioinformatician. There are a lot of data, genomic tools, proteomic tools, venom pharmaceutical tools, and structural biology tools like that. You have a lot. And one of the tools or one of the suite present in XPC is called Swiss Tools or Swiss Suite. So SIB, Swiss Institute of Bioinformatics, is the owner of this particular framework. So in this, you can do docking in your software, docking in your web browser, and you can find similar targets. You can find similarities between chemical compounds. You can just construct molecules like that, and you can do pretty much more like all this proteomic stuff and all this chemical related analysis. You can do it here. And PDB is the secondary database for protein. You have learned about this in your courses maybe. Drug bank is where when a drug is published or there is a drug, like for example paracetamol, aspirin. So if you wanted to have a full set of information of that particular drug, the drug likeness, the ADMET profiling, the characteristics of the drug, physiochemical characteristics, the interaction of that particular drug, the chemical markup of the drug. You can just visit this drug bank and you can analyze any drug. If it is in a drug form, if it is repurposed or published as a drug, you can just go and check there. Or if it is a generic chemical molecule, just go and check on PubChem. If you're coming across any kind of IUPAC nomenclatured chemical, or any generic kind of chemical which is not identified or just identified using your HPLC or GCMS, you can just put the data, you can search for the data in PubChem. Okay, so this is the end. So I guess today we have discussed briefly about the introduction. So the forum is open for questions. You can ask queries. So you can ask questions. KM Ashwani, you raised your hand. Yes, you guys have questions? Any queries? Anyone? Guys, am I audible? Like, you guys can hear me?
Yes, sir, you are audible.
Yes, sir. Yes, sir.
Okay, sorry. I thought, yeah, tell me if you have any questions or if you wanted to show something, I just can just discuss about that. Excuse me. So none. Since this is an introduction class, you guys don't have any uh, questions. Is that it?
Sir, can you explain the role of workflow management tool?
Workflow management tool? Yeah, sure. Just a minute. I'll just show you. So can you guys see my screen? Is my screen visible?
No, sir.
Just a minute. Just a minute. So now, is it open? You guys can see. Can you see my screen? Yes.
Yes, sir.
Yes, sir.
Yes.
Yes.
Yes, now.
So we talked about this Galaxy, right? So Galaxy is an open source web-based platform where you can analyze this biological data and you can construct workflow. So in the left-hand side, you can see just what are the options there. So genomic file management, manipulation. So it's just a collection of or repository of data, or sorry, collection of tools that you can automate or you can do within this particular workframe. So you can analyze FASTA and FASTQ files and FASTQ quality control. FASTQ is the file format when you when it is given after a NGS, next generation sequencing analysis. So when you going for NGS, next generation sequencing, the file format is which is given by the vendor or the provider will be FASTQ file. So next thing will be constructing alignment files, SAM and BAM, and we'll be checking for its efficacy, its quality, and like that. And common genomic tools are here, and genomic assembly tools, multiple alignment tools like that. So these all tools are here where we can just automate a model or automate a workflow from here. No need to, like for example, you can just download a tool, you can install it, you can do a work, and it will give you result, right? And that is not much needed because you have a lot of tools baked inside this workflow. Okay, a lot of tools baked inside this workflow where you can these particular tools are available outside the web where you can download and use. But here you don't need to download the software, but you can just use inside the workflow work frame itself. Okay, here you can visit this website and can just have a look at it. What all you can do. Okay, you can, and this is the website, and you can see their newsletters and stuff. Okay, so I didn't automate any workflow, so my uh, that's why my history is nil. So this is one of the workflow. If I wanted to show you some workflow, I should prepare some prerequisites. So in that case, I'll construct a workflow, and I should be having some prerequisites, right? So I'll just show you when we are discussing about this NGS data analysis. That is next chapter. That is next topic. Okay. Is that okay? And I'll show you by then. Is that fine?
Okay, sir. Okay, sir.
Yes. So, um, and anything else?
Sir, will there be any exam at the end of this course?
Regarding your test and exam, I'm not clear because I'm just a trainer. Okay, and I'm getting information that you'll be having exams. I am not sure about it. Sorry. I'll check and let you know. Okay, regarding the test or any sort of examinations if you're having some MCQs. If in that case, we'll let you know. Okay,
Okay, sir.
for your qualification. Yes. Anything else?
Sir, I had a question.
Yes, Amit, tell me.
So basically, I went to Upwork to see the types of work being done on like the you know bioinformatics
Okay,
jobs,
okay.
like freelancer jobs. So I wanted to know if there is a repository or a place where we can create, we can get ideas for making projects that we can showcase to potential,
For being a freelancer, to
yeah, for
to be a
yeah,
freelancer. So, were you there yesterday?
huh?
Were you there? Were
Yeah,
you present
yeah,
yesterday?
yeah, yeah. I
Yeah,
was
that's
there.
great. So I told you about my stuff, right? So I just started like helping my friends, okay? And
Okay,
I also checked and I got registered in a freelancing domain. Just
okay,
forgot the domain's name, like similar to Upwork. Okay, so you can create a
okay,
profile there, and you can just provide your freelancing services there. And one more thing, you can just help your friends or help other people. And there are some WhatsApp groups and channels and forums like that. Okay, so that you can give an advertisement. You should be having a proper designed, well-defined and well-tabulated kind of brochure for yourself. Okay,
okay,
this is not like you should have a company to do this. You're just doing under your name. You're free to do. Okay, you can create a brochure defining your process. When where which all areas you can just help the people who are doing this in vitro and stuff. Okay, and you
okay.
can just charge them. You can put a charge table and you can just charge them. Okay, so
Yes.
this is how I constructed a brochure, and I shared it across some WhatsApp groups among my fellow mates, among my juniors and students. And also, I took help of my faculty. They shared this particular brochures to the people. This is how this network develops because we are not business-minded people, right? We're just freelancing for stuff. So this is how it developed. And I'll just check and let you know what is the website that I uh, registered.
Okay.
So, so in that case, what would be your idea on freelancing? Like, what will you be doing?
Sir, I want to work with Python, but I'm not sure what kind of projects that I should showcase for
Yeah,
bioinformatics.
that is great.
So
So, you can register yourself in Fiverr. You know
Fiverr.
Fiverr? Fiverr?
Yeah, yeah,
So
yeah.
just go there, just search for some projects. Okay, similar kind of as you're going to verify some projects. Like as you need some service from Fiverr, just go and search like that. Okay,
Okay,
so you get an idea. Basically, Python freelancing on bioinformatics totally relies on data visualization, where you need to process the raw data and give them the plot data, graphical pie charts like that. Okay,
okay,
this is one of the freelancing you can do. And next thing is automation of a workflow. Automation of a workflow, you can create an algorithm or create a .py executable file where
yeah,
they can just execute in their local system. You can construct a program like that where it does the job. For example,
yeah.
uh, I'll just show you some. So you guys could see my screen. So I searched for BioPython, my browser. So I already had this notebook. This is the BioPython notebook. So what are the programs you can run? I created this notebook for a different class actually. So what all tools you can do is you can see here. So this particular tool, let me tell you, let me explain you the easiest one. So this particular tool is the tool which takes the input of a user and converts the input, in the sense you will be giving the raw data, the sequence data, and it will be finding the complementary and reverse complementary of that particular DNA sequence. So it's just a 4 line to 5 line kind of Python scripting. Okay, like you can see, these are simple tools, simple kind of small tools. Like for pairwise element, we can create a tool. For multiple sequence element, we can create a tool. Okay, for converting the sequences, or for post translational or protein translation from RNA, you can create some tools. So this is for automating these kind of processes. You can create uh, some kind of um, these kind of programs, and you can just provide that program to them. Okay, or else what you can do is you can just register yourself in GitHub, and you can deposit this in the repository, in the private repository, and you can give the access to the people. Like that you can do. Okay, so these are small examples. It's not big kind of codings what I did. These are small examples. Okay, so this is what I talked about. This is Google Collaboratory, like Google Colab, where you can do coding and executing that code. You can execute those codes in the browser. Okay. So is that okay, Amit?
Yes, sir.
So regarding those, I can do automation. Even some people, they wanted to do automation for molecular docking and molecular simulation. For that, you can construct a program with a single click of a button. It can just take up the inputs of the user, and it can give the data. Like that, you can construct some programs. And if you're not well versed in programming stuff, you can always reach out to GitHub repository. There are a lot of sample codes. You can check on BioPython. You can ask the help of ChatGPT. ChatGPT is for the primary screening, or primary set of uh, just for checking. So there are other ChatGPT models where you can get the aid of itself for constructing a code, constructing the scripts actually. Okay, so freelancing in Python with bioinformatics is, this can be in such a way, designing workflows and automating the workflow. Anything else?
No, sir. Thank you so much.
Yeah, thank you. So just check on Fiverr. Okay, there's a website. This is the website, and this is the website where you can find the sources. Sorry, that is freelancing web sources. So I'll put in the chat box. You can just view. Okay. So anyone else? You guys have any questions? So no other questions. We'll end the session. Okay. Someone raised your hand. Okay, no. So in that case, if you guys don't have any question, we'll conclude the session. And tomorrow we'll start with this next thing, that is next generation sequencing data analysis. We'll be talking about pre-processing and quality control of NGS data, and alignment and mapping of the sequence reads, and variant calling and structural analysis, structural variant analysis calling, and what are the tools we're going to use for this NGS data analysis. So we'll discuss about that in the like in tomorrow's class. Okay. Yeah. So PPT, I'll share it in the WhatsApp group. You can just download from there. Not like a link. I'll just share it as a file itself. And there's my the first page. I've given my LinkedIn ID. You can just go and check it. And if at all you have any doubts, you can just reach out and DM me. Okay. So let's conclude the session.
Thank you, sir.
Yes. Yeah. Thank you so much. Thank you all for listening.
Thank you, sir.
And yeah, just go through the slides, and if you have any doubts, we'll discuss tomorrow. Yeah. It's happy learning. Thank you so much.
Thank you, sir.
Thank you.